Consider for a moment the profound paradox of your existence, where the chaotic entropy of the universe meets the persistent, organized renewal of the biology of aging. It is a quiet phenomenon. Within the intricate architecture of your cells, a constant struggle unfolds between the inherent drive to maintain order and the inevitable accumulation of molecular noise. This process, formerly known as senescence, is not merely a linear decline or a simple wearing down of parts, but a highly coordinated biological symphony that responds to internal and external pressures. Every second, trillions of chemical reactions sustain your vitality, yet each one leaves behind a microscopic footprint, a tiny signature of time passing. This transformation reaches from the smallest subatomic particles and the fraying of DNA strands to the grand shifts in how societies are structured around longevity. We often rely on the steady rhythm of a calendar to measure our lives, but your body maintains a different ledger altogether. This is the critical distinction between your chronological age, the number of years you have occupied space on this planet, and your biological age, which reflects the actual physiological state of your tissues and organs. While the calendar moves at a fixed velocity, your biological clock can accelerate or decelerate based on a complex interplay of genetics, environment, and cellular metabolism. It is a fascinating reality that two people born on the same day can be decades apart in terms of cellular health. If you find these insights into our inner workings helpful for your bedtime listening, perhaps you might subscribe to join our quiet community of curious minds. The mechanism of aging is fundamentally rooted in the loss of information. As cells divide, the protective caps on the ends of chromosomes, called telomeres, gradually shorten like the burning wick of a candle. When these caps become too short, the cell enters a state of permanent arrest, no longer contributing to the repair of the body but instead secreting inflammatory signals that affect neighboring cells. This ripple effect contributes to the gradual thinning of skin and the slowing of wound healing. Interestingly, research published in the journal Nature suggests that certain organisms, like the Greenland shark, possess unique genetic adaptations that allow them to resist these processes for centuries. Do you remember a time when you first noticed a subtle change in your own physical endurance, or perhaps the way your skin felt in the morning? Such observations are our first personal encounters with the underlying mechanics of senescence. This journey into the biology of time reveals that we are not just victims of the clock, but active participants in a deeply complex and still largely mysterious evolutionary program designed to balance life and renewal. To understand why our cells eventually falter, we must look toward the fundamental laws of physics that govern the entire universe. The second law of thermodynamics dictates that entropy, or the measure of disorder, must always increase within a closed system. Life represents a temporary and defiant rebellion against this inevitable decay by constantly importing energy to maintain an internal state of high organization. Every heartbeat and every synaptic spark serves as a bulwark against the chaos that seeks to dissolve our structural integrity. This process is not free, as the body must burn fuel simply to stay the same, a concept often described in physics as maintaining a steady state far from equilibrium. The metabolic cost of this resistance is immense, requiring a constant flow of nutrients to power the molecular machinery of repair. Inside every cell, specialized enzymes act like a microscopic maintenance crew, patrolling the vast strands of DNA to fix breaks and errors caused by radiation or chemical stress. However, no mechanical or biological system achieves perfect efficiency, and a small fraction of the energy we consume produces harmful byproducts known as free radicals. These unstable molecules act like sparks flying from a forge, occasionally striking and damaging the very structures they were meant to support. Over several decades, these tiny, localized failures begin to aggregate into a significant burden of cellular noise that interferes with healthy function. As we move through the middle decades of life, the accumulation of these errors starts to outpace the body's ability to rectify them. Think of it as a long-term transcript that has been photocopied thousands of times. Eventually, the clarity of the original message is lost to the blur of repetition. This loss of information at the cellular level is a primary driver of the physical changes we associate with growing older. While the body is remarkably resilient, it is not designed for infinite durability, but rather for a specific window of peak performance. Do you remember a time when you felt your body could recover from almost anything overnight? Evolutionary biology suggests that our internal resources are distributed according to a strict hierarchy of survival needs. The disposable soma theory, popularized by biologist Thomas Kirkwood, proposes that the body is essentially a vehicle for the germline cells that carry our genetic legacy forward. Because resources like energy and nutrients are often scarce in nature, organisms must make a calculated trade-off between maintaining their current body and investing in the next generation. Once an individual has reached reproductive maturity and potentially passed on their genes, the evolutionary pressure to maintain the body with high-fidelity repair mechanisms begins to wane. This shift in biological priority means that the energy once used for pristine cellular upkeep is gradually diverted or simply reduced. Nature prioritizes the continuity of the species over the eternal life of the individual, leading to a slow but deliberate withdrawal of metabolic investment. We find ourselves at the mercy of a program that views our post-reproductive years as a period of diminishing returns. This strategic abandonment sets the stage for the complex evolutionary pressures that have shaped the maximum lifespan of every creature on Earth. Evolutionary biologists refer to our physical forms as the disposable soma, a concept suggesting that the body is merely a temporary vessel designed to ensure the survival of our genetic information. Within this framework, resources are limited, and an organism must decide whether to invest energy into maintaining its own tissues or into the act of reproduction. Once an individual has successfully passed on its genes, the evolutionary pressure to maintain the body begins to fade, as the survival of the species is already secured in the next generation. This trade-off implies that our inevitable decline is not a mistake of nature, but a calculated prioritization of the future over the present. This logic extends into the theory of antagonistic pleiotropy, which suggests that certain genes possess a dual nature that changes over the course of a lifetime. A gene might offer a significant advantage during our youth, such as enhancing calcium metabolism for bone growth or boosting the immune system to fight off early infections. However, those same genetic instructions can become detrimental in our later years, perhaps causing the calcification of arteries or chronic inflammation. Because evolution prioritizes the period of life where we are most likely to reproduce, these late-life disadvantages are never filtered out of the gene pool. Do you ever find yourself wondering if the boundless energy of your youth was actually a loan taken against your future health? While some genes are actively harmful in old age, others simply slip through the cracks of natural selection via a process known as mutation accumulation. Evolution acts like a high-pressure sieve, catching and removing mutations that kill or sterilize the young before they can reproduce. But for mutations that only manifest their symptoms after the reproductive window has closed, the sieve is much coarser. These late-acting genetic errors accumulate over thousands of generations because there is no biological mechanism to penalize them. We are, in a sense, a collection of genetic glitches that simply didn't show up early enough to be stopped. The natural world is filled with examples of this preference for early vigor over long-term stability. The Pacific salmon, for instance, undergoes a rapid and total physiological collapse immediately after spawning, a process driven by a massive hormonal surge that ensures successful mating at the cost of the individual's life. While human aging is far more gradual, the underlying principle remains that longevity is rarely a primary objective for the forces of selection. If a mutation allowed an animal to live twice as long but slightly reduced its fertility in its prime, that mutation would almost certainly disappear from the population. There are, however, fascinating anomalies that suggest living long past reproduction might hold its own hidden evolutionary value. The grandmother hypothesis proposes that postmenopausal longevity in humans evolved because elder females could provide essential care and food for their grandchildren, thereby increasing the survival rates of their descendants. This suggests that while our individual bodies are disposable, our presence as providers of wisdom and stability can still influence the success of our genetic lineage. This shift from physical vigor to social contribution marks a rare moment where evolution seems to value the endurance of the individual. If you find these insights into our biological architecture peaceful to contemplate, you might consider following the channel for more science for sleep. Every year we live is a testament to a complex balance between the drive to persist and the necessity of making way for what comes next. In the early 1960s, a microbiologist named Leonard Hayflick at the Worcester Institute overturned decades of biological dogma regarding the nature of cellular longevity. Until his meticulous experiments, the scientific community largely followed the theories of Nobel laureate Alexis Carrel, who asserted that vertebrate cells were inherently immortal if provided with the correct nutrients. Hayflick demonstrated that normal human fetal cells actually possess a sophisticated internal counting mechanism that strictly limits their ability to divide. This boundary, which we now recognize as the Hayflick limit, typically restricts a cell population to between 40 and 60 doublings before the process permanently halts. Do you remember first learning about the basic building blocks of life in school and wondering how they knew exactly when to stop growing? When a cell reaches this predetermined threshold, it enters a state known as replicative senescence. During senescence, the cell remains metabolically active and resists programmed death, yet it loses the capacity to undergo further mitosis. This phenomenon serves as a fundamental barrier against the uncontrolled growth characteristic of cancer, acting as a biological safeguard for the organism. However, this same safety mechanism contributes to the gradual decline in tissue regeneration that defines the aging process. The accumulation of these non-dividing cells can eventually lead to chronic inflammation and the degradation of the surrounding extracellular matrix over many decades. This finite lifespan is a specific characteristic of somatic cells, which constitute the vast majority of the human body's structure and function. In contrast, germ-line cells, those responsible for reproduction, along with certain types of stem cells, are capable of bypassing these limitations to ensure the continuity of the species across generations. While somatic cells are tethered to a mortal timeline, the germ line maintains a kind of biological immortality, passing on genetic information without the same degradative constraints. This distinction highlights a profound evolutionary trade-off between the survival of the individual and the persistence of the lineage. The reason for this strict mathematical limit remained a mystery for years until researchers looked closely at the physical ends of our chromosomes. Each time a cell prepares to divide, it must replicate its entire genome, but the enzymes responsible for this duplication cannot reach the very tip of the DNA strand. This inherent limitation in the copying process means that a small fraction of genetic material is lost with every single division. To protect the vital coding sequences from being eroded, our chromosomes are capped with repetitive, non-coding sequences of DNA known as telomeres. These protective buffers act like the plastic tips on shoelaces, preventing the genetic thread from fraying during the cycle of replication. Each time a cell replicates its genetic blueprint, the enzyme responsible for copying the DNA, known as DNA polymerase, faces a structural limitation. Because it can only synthesize new strands in one specific direction and requires a pre-existing primer to begin, it cannot replicate the very tip of the linear chromosome. This is commonly referred to as the end replication problem, a biological quirk that would result in the loss of vital genetic information if not for the presence of telomeres. These repetitive TTAGGG sequences act as a sacrificial buffer, allowing the cellular machinery to shave off a small portion of the cap rather than the essential genes located further down the strand. Over the course of a human lifetime, these caps inevitably shrink, serving as a molecular clock that tracks the age of a specific cell lineage. When the telomeres reach a critically short length, the cell enters a state of permanent growth arrest called senescence, or it may undergo programmed cell death. This limit on cellular division is a primary driver of the physical manifestations of aging, yet nature has developed a way to bypass this countdown under specific circumstances. The enzyme telomerase serves as a specialized architect, capable of adding DNA back onto the ends of chromosomes to maintain their length and integrity. In 2009, Elizabeth Blackburn, Carol Greider, and Jack Szostak were awarded the Nobel Prize in Physiology or Medicine for discovering how telomeres and the enzyme telomerase protect our chromosomes. While telomerase is highly active in embryonic stem cells and germ cells, ensuring that the blueprint for the next generation remains pristine, it is largely silenced in most adult somatic cells. This silencing is thought to be an evolutionary trade-off designed to suppress the development of cancer, as uncontrolled cell division requires a way to maintain telomere length indefinitely. Interestingly, nearly 90% of human cancer cells find a way to reactivate telomerase, granting them a form of biological immortality that allows tumors to grow without the constraints of the Hayflick limit. Do you ever wonder if the stresses of your daily life are being etched into your very DNA at this moment? Scientific research has demonstrated a profound link between chronic psychological stress and accelerated telomere shortening. When the body is flooded with cortisol and oxidative stress markers, the rate of telomeric attrition increases, effectively aging the body's cells faster than the calendar would suggest. Environmental factors, including diet, sleep quality, and exposure to pollutants, also play a decisive role in how quickly these protective caps erode. While we cannot yet stop the clock entirely, maintaining a lifestyle that supports cellular health can significantly influence the persistence of these chromosomal buffers. The study of telomere dynamics reveals that aging is not merely a passive wearing down of the body, but a highly regulated, interactive process between our genetics and the world we inhabit. As these caps slowly diminish, they signal the transition from the vibrant energy of youth to the measured stability of maturity. If DNA represents the rigid hardware of our biological computer, the epigenome acts as the sophisticated software directing which programs run and when. While our genetic sequence remains largely fixed from the moment of conception, the epigenome is a fluid, reactive layer of chemical modifications that dictates how our cells interpret those instructions. This layer ensures that a heart cell and a skin cell can share the exact same blueprint while performing vastly different functions, simply by silencing certain chapters of the genetic code and highlighting others. At the heart of this regulatory system is a process called DNA methylation, where small chemical clusters known as methyl groups attach to specific locations on the double helix. These tags do not change the genetic letters themselves, but they act like molecular dimmers, silencing specific genes or allowing others to express themselves with high intensity. As we move through the decades, the pattern of these tags shifts in a predictable, rhythmic fashion that reflects our true internal status. This shift is not random. It is a systematic drift that characterizes the transition from the resilience of youth to the vulnerability of advanced age. This predictable progression led to the revolutionary development of the Horvath clock, a mathematical model that measures biological age by analyzing methylation levels across hundreds of sites in the genome. Unlike a calendar, which only counts the revolutions of the Earth around the Sun, this epigenetic clock provides a high-resolution look at the actual wear and tear on our cellular machinery. By observing these chemical signatures, researchers can often predict health outcomes and functional decline more accurately than a simple birth date ever could. Have you ever noticed how two people born in the same year can seem to exist at entirely different stages of their physical journey? Our understanding of this clock has moved beyond mere observation into the realm of active intervention, particularly following the breakthroughs of 2024 and 2025. During this recent period, pioneering clinical trials demonstrated that it is possible to reboot certain epigenetic markers, essentially clearing away the molecular noise that accumulates over time. Scientists are now utilizing transient cellular reprogramming, a technique that briefly activates specific factors to refresh the epigenome without erasing the cell's fundamental identity. These 2025 advancements have confirmed that cellular aging is not an inevitable decay, but rather a loss of information that can, in some cases, be restored to its original, clearer signal. Every choice we make serves as a pen that writes onto this epigenetic ledger, creating a bridge between our environment and our internal biology. Factors such as the quality of the air we breathe, the depth of our nightly rest, and the specific nutrients in our diet send signals that tell the epigenome which genes to lock away and which to keep active. Chronic stress has been shown to accelerate the accumulation of aging methyl tags, while regular physical activity helps maintain a more youthful epigenetic profile. Do you find it empowering to think that your daily choices are whispering instructions directly to your genome? These environmental interactions ensure that we are not merely passive victims of our inheritance, but active participants in the unfolding story of our own longevity. This dynamic interplay means the clock of aging is not a fixed countdown, but a complex mechanism that we are finally learning how to pace. Deep within the architecture of every cell, microscopic power plants known as mitochondria work ceaselessly to convert the oxygen we breathe and the food we eat into adenosine triphosphate, or ATP. This molecule serves as the primary currency of biological energy, fueling everything from the blink of an eye to the complex firing of neurons in the brain. As we age, these essential organelles begin to lose their efficiency, creating a fundamental shift in our internal vitality. The free radical theory of aging, first proposed in the mid-20th century, suggested that the reactive oxygen species produced as a byproduct of this energy creation slowly corrode the cell from the inside out. Modern research, however, offers a more nuanced perspective, showing that low levels of these radicals might actually serve as vital signaling molecules that trigger protective cellular responses. It is when the production of these molecules overwhelms the cell's antioxidant defenses that the delicate internal machinery begins to suffer. Does the feeling of a particularly deep, restorative sleep ever make you wonder how much energy your cells are reclaiming in the stillness? This question touches on the heart of metabolic health, as the decline in ATP production is one of the most consistent markers of the aging process across species. Unlike the primary DNA found in the nucleus, mitochondrial DNA lacks the robust protection of histone proteins and is physically closer to the site where damaging free radicals are generated. Over decades, mutations accumulate within this circular genome, leading to a structural breakdown that impairs the cell's ability to generate heat and power. To combat this, the body employs a sophisticated quality control mechanism called mitophagy, a specialized form of autophagy that identifies and digests broken or dysfunctional mitochondria. In a youthful state, this recycling process is vigorous, ensuring that the cellular population of power plants remains healthy and efficient. With the passage of time, the signals that trigger mitophagy often weaken, allowing damaged mitochondria to linger and take up space without contributing to the energy pool. This accumulation of cellular clutter is a hallmark of biological aging, contributing significantly to the persistent fatigue and reduced physical stamina often associated with later life. When the mitochondrial network fragments and becomes less interconnected, the metabolic flexibility of the entire organism begins to shift. Cells that were once adept at switching between different fuel sources, like glucose and fatty acids, become rigid and less responsive to the body's demands. This metabolic inertia can lead to systemic changes, including altered insulin sensitivity and a decrease in basal metabolic rate, which influences how we store and utilize nutrients. The preservation of mitochondrial health through specific lifestyle interventions remains one of the most studied areas of longevity science in 2026. By maintaining the integrity of these ancient organelles, we are essentially protecting the very spark that keeps the biological machine running. Science now looks toward therapies that can stimulate mitochondrial biogenesis, the birth of new power plants, to offset the natural decline of the old ones. The concept of cellular senescence describes a biological state where cells permanently cease their division cycles while resisting the natural process of programmed cell death. Instead of quietly exiting the stage through apoptosis, these biological actors linger in a state of suspended animation, earning them the evocative nickname of zombie cells. This phenomenon was first characterized by Leonard Hayflick in the early 1960s, identifying a finite limit to how many times a human cell can replicate before its telomeres become critically short. When a cell detects significant DNA damage or structural instability, it enters this senescent state to prevent itself from replicating errors that could potentially lead to malignancy. In this context, senescence is a heroic defense mechanism, acting as a primary barrier against the development of cancer during our younger, more reproductive years. As the decades pass and we move through 2024 and 2025, the accumulation of these dormant cells within the tissues of the heart, brain, and skin shifts their role from protectors to primary contributors of systemic decline. The primary vehicle for this damage is a complex chemical profile known as the senescence-associated secretory phenotype, or SASP. This term refers to the potent cocktail of signaling molecules, including pro-inflammatory cytokines, growth factors, and tissue-wasting enzymes, that senescent cells continuously broadcast into their immediate surroundings. Have you ever noticed how a small area of skin takes just a little longer to heal than it did in your earlier years? This persistent chemical broadcast creates a microenvironment of chronic, low-grade inflammation, a state that researchers now frequently categorize as inflammaging. This evolutionary trade-off, where a trait provides a survival benefit early in life but exacts a heavy cost later on, is known as antagonistic pleiotropy and remains a cornerstone of modern gerontology. As these zombie cells persist, they take up physical space and consume metabolic resources without contributing to the actual function of the organ, effectively acting as a drag on the entire body's biological efficiency. Fortunately, our internal systems possess a primitive but highly effective garbage disposal mechanism designed to handle cellular debris and dysfunctional components. This process, known as autophagy, allows the body to identify, break down, and recycle the internal parts of a cell that are no longer performing their duties correctly. When the rate of senescence begins to outpace the body's natural ability to clear these cells through autophagy and immune surveillance, the physiological scales tip toward the visible and invisible markers of aging. Science is now intensely focused on finding ways to enhance this internal cleaning or to selectively eliminate these stubborn residents through a class of emerging compounds that allow healthy cells to thrive without the shadow of constant inflammatory interference. If you find these biological mysteries as fascinating as I do, perhaps you'll consider subscribing to stay connected with our future explorations. Within the microscopic landscape of your cells, a silent and continuous process of origami determines the very foundations of your health. This is the realm of proteostasis, a sophisticated system of quality control that ensures every protein is folded into its exact three-dimensional shape. Each protein starts as a linear chain of amino acids, but it only gains its power once it twists, curls, and snaps into a specific configuration. This shape is not an aesthetic choice. It is a functional necessity. If a protein is folded incorrectly, it cannot dock with its intended targets, rendering it useless for the vital tasks of life. When these proteins misfold, often due to the cumulative stresses of heat, oxidation, or genetic errors, they do not simply disappear. Instead, they tend to expose sticky surfaces that would normally be tucked away in a healthy state. These rogue molecules begin to find one another, clumping together into dense, insoluble masses known as aggregates. You may know these as the plaques and tangles that are often cited in discussions of cognitive decline. These aggregates act as physical barriers within the delicate circuitry of the cell, disrupting the flow of nutrients and the transmission of electrical signals that define our consciousness. To combat this accumulation of biological clutter, the body employs a remarkable survival mechanism known as autophagy, which literally translates to self-eating. This is your internal recycling program, a meticulous janitorial service that scans the cytoplasm for damaged organelles and malformed proteins. Once identified, this waste is wrapped in a double-membrane bubble called an autophagosome and transported to the lysosome. Think of the lysosome as a tiny, acidic furnace where the waste is dismantled into its original amino acids, providing the raw materials for the cell to rebuild itself from within. This process is a core focus for those interested in science for sleep, as it highlights the restorative nature of our biology. As we navigate the later decades of life, the efficiency of this cleanup crew naturally begins to wane. The signaling pathways that tell the cell it is time to take out the trash become muffled, and the mechanical components of the autophagy machinery start to wear down. This decline in proteostasis means that the cellular environment becomes increasingly crowded with garbage that should have been recycled long ago. This sluggishness is one of the primary reasons aging is associated with a loss of vigor at the cellular level, as the energy once used for growth is redirected toward managing this mounting internal stress. This failure of protein management creates a direct bridge to neurodegenerative conditions. In a healthy young brain, the turnover of proteins is swift and thorough, but in an aging brain, the accumulation of these toxic proteins can eventually overwhelm the system. By understanding the intricate dance of folding and recycling, researchers are finding new ways to potentially boost these natural defenses. If we can maintain the integrity of our cellular origami, we may be able to preserve the clarity of the mind well into our later years. This delicate balance of creation and destruction is what allows us to endure, a constant renewal that defines the very essence of living. Within the intricate architecture of our tissues lies a specialized population of cells known as adult stem cells, which act as a biological repair crew. These undifferentiated laborers possess the unique ability to divide and transform into the specific cell types required to replace those lost to injury or natural wear. Throughout our youth, this regenerative engine operates with remarkable efficiency, ensuring that the lining of the gut is replaced every few days and the skin remains a robust barrier. As the decades pass, the reservoirs of these cells begin to dwindle in a process scientists call stem cell exhaustion. It is not always that the cells vanish entirely. Often, they enter a state of deep dormancy known as quiescence, becoming unresponsive to the chemical signals that normally trigger repair. This gradual decline in the progenitor pool means that the body's capacity for self-renewal slows down, leaving gaps in the cellular mosaic that were once filled instantly. In the bone marrow, the hematopoietic stem cells that produce our blood encounter their own hurdles as we age. Their ability to generate a diverse array of immune cells shifts, often favoring the production of myeloid cells over lymphoid cells, which can weaken our adaptive immune response. This shift is a primary reason why older individuals may become more susceptible to new pathogens or find that vaccines are less effective than they once were. The impact is equally visible in our physical form, where the slowing of skin stem cells leads to thinner tissue and delayed wound healing. In our muscles, the satellite cells responsible for repairing fibers after exertion lose their vigor, making it harder to maintain strength even with consistent activity. Have you ever noticed how a small bruise or scratch seems to linger much longer than it did when you were a child? Beyond just the number of cells available, the quality of the remaining stem cells undergoes an internal decline characterized by DNA damage and epigenetic shifts. The very instructions they carry become frayed, leading to stem cell aging where the daughter cells they produce are less functional than their predecessors. Research published in journals like Nature suggests that this exhaustion is one of the primary drivers of systemic decline across almost every organ system. This internal depletion eventually manifests in the mirrors of our daily lives, where the microscopic failure to replenish translates into macroscopic change. The structural integrity of the body begins to soften, not because of a single catastrophic event, but because of a trillion tiny absences. As the regenerative capacity reaches its limit, the cumulative effect of these missing pieces begins to alter our very appearance and vitality. Beyond the individual cell membrane lies a complex scaffolding known as the extracellular matrix, a non-cellular framework that dictates the physical resilience of every tissue in the body. This matrix is far more than a simple filler. It is a dynamic, interconnected network of fibrous proteins and carbohydrates that coordinates the behavior of the cells it cradles. Collagen serves as the primary structural component of this environment, providing the tensile strength necessary for skin, bone, and vascular integrity. As the years pass, the fibroblasts responsible for synthesizing these proteins gradually lose their vigor, resulting in a measurable thinning of the structural lattice. Have you ever noticed how the texture of a leaf changes as it dries, losing that supple quality it held in the spring? This shift in human biology is driven by the fragmentation of elastin, the protein that allows tissues to snap back into place after being stretched or compressed. When elastin fibers break down, the skin begins to follow the relentless pull of gravity, leading to the characteristic sagging observed in later life. The integrity of this matrix is further compromised by a chemical process known as glycation, where circulating sugar molecules bind spontaneously to protein fibers. These sugar-protein bonds create advanced glycation end products, or AGEs, which act like microscopic glue, cross-linking once flexible collagen strands into rigid, unyielding bundles. This stiffening of the extracellular environment creates a physical barrier that hinders the efficient diffusion of oxygen and vital nutrients from the bloodstream to the cells. In the cardiovascular system, this loss of elasticity forces the heart to pump against stiffer arterial walls, a primary factor in age-related increases in blood pressure. The matrix also serves as a reservoir for growth factors, but as it densifies, these signals become trapped or distorted, confusing the regenerative instructions sent to the organs. While these changes occur naturally over time, the environment plays a decisive role in the speed of the transition. Ultraviolet radiation is particularly destructive, as it triggers the overproduction of enzymes called matrix metalloproteinases that aggressively dismantle the skin's structural support. This process, known as photoaging, creates a stark contrast between the protected skin of the inner arm and the weathered areas frequently exposed to the sun. Even the hydrating molecules of the matrix, such as hyaluronic acid, begin to diminish, reducing the tissue's ability to retain moisture and maintain its youthful volume. This gradual dehydration of the interstitial space contributes to the visible fine lines that mark the passage of decades. As the framework of the body loses its fluidity, the very architecture of our physical form undergoes a fundamental reorganization that mirrors the slowing of our internal biological clocks. This structural decline is further complicated by a pervasive biological phenomenon known as chronic, low-grade inflammation. While acute inflammation is a vital, life-saving response to injury or infection, marked by heat, redness, and swelling, the aging body begins to simmer with a different, quieter fire. Scientists refer to this as inflammaging, a sterile form of inflammation that persists even in the absence of an external pathogen. Unlike the temporary surge of activity required to heal a wound, this persistent state acts as a background noise of cellular stress that never truly settles. Over time, the metabolic cost of maintaining this heightened state begins to erode the very tissues the immune system is meant to protect. The innate immune system, our body's first line of defense, is the primary driver behind this escalating tension. As we age, the cells responsible for identifying threats become hyperreactive yet less efficient at clearing away the debris of cellular living. This results in a state of constant, low-level mobilization that gradually damages healthy cells. Have you ever noticed how your body seems to take longer to recover from even the smallest physical strain than it did years ago? This lingering sensitivity is often the outward shadow of an immune system that has lost its ability to return to a baseline of calm. One of the most fascinating triggers for this internal alarm system is the presence of misplaced biological material, such as fragments of DNA. Normally, DNA is securely housed within the nucleus or the mitochondria, but as cells age and experience damage, these genetic strands can leak into the cytoplasm. The immune system misidentifies this self-material as a foreign invader, like a virus, and launches a defensive strike against its own host. This accumulation of cellular junk, which also includes misfolded proteins and damaged organelles, provides a constant source of fuel for the inflammatory fire. Research published in Nature Aging suggests that this molecular debris is a fundamental cornerstone of the aging process across various species. This persistent inflammatory state is not merely a side effect of growing older. It is a central pillar that connects almost all age-related pathologies. From the stiffening of arterial walls in cardiovascular disease to the cognitive decline associated with neuroinflammation, the thread of inflammaging runs through the most common challenges of the later years. It creates a feedback loop where inflammation causes tissue damage, and that damage, in turn, recruits more inflammatory signals. By understanding this invisible chemical landscape, researchers are beginning to view aging not as a series of unrelated failures, but as a single, systemic shift in how our bodies manage internal harmony. This shift represents a transition from a period of growth and repair to one of maintenance and, eventually, a struggle against the entropy of our own defense mechanisms. As the signaling molecules known as cytokines circulate through the blood, they carry messages of urgency that the body can no longer satisfy, leading to a gradual exhaustion of our biological reserves. This exhaustion marks the move from the structural changes of the body toward the deeper, more complex world of genetic instability and the secrets held within our telomeres. Within the vault of the skull, this exhaustion manifests as a physical sculpting of the landscape where our memories and identities reside. The brain begins to lose volume in a process known as cerebral atrophy, a phenomenon that typically accelerates after the age of 60. We see the most pronounced thinning in the prefrontal cortex, the seat of executive function, and the hippocampus, which serves as the primary gateway for the formation of new memories. It is here that the structural architecture yields to time, with individual neurons shrinking and the complex branching of dendrites becoming significantly less dense. This reduction in the arborization of neurons means that each cell has fewer points of contact with its neighbors, subtly altering the network's efficiency. As the gray matter recedes, the internal fluid-filled cavities known as ventricles gradually expand to occupy the vacated space. This change is accompanied by a subtle but significant degradation of white matter, the lipid-rich insulation that allows electrical impulses to travel rapidly between distant brain regions. When the integrity of these myelinated tracts falters, often due to small-scale vascular changes or the loss of oligodendrocyte support cells, the processing speed of the mind begins to mirror the slower pace of the aging body. Can you recall a moment when a word was on the tip of your tongue, only to surface minutes later after the conversation had already moved on? This delay is a tangible reflection of the lengthening transit times for signals traversing the cerebral landscape as they navigate these microscopic structural detours. The protective infrastructure of the brain also faces challenges, specifically within the blood-brain barrier. This semi-permeable border is designed to shield sensitive neural tissue from toxins, inflammatory molecules, and pathogens circulating in the bloodstream. Over decades, the tight junctions of the endothelial cells forming this barrier may loosen, a state of increased permeability that researchers often link to chronic neuroinflammation. As these defenses weaken, the internal environment of the brain becomes more susceptible to metabolic fluctuations and protein misfolding, factors that can disrupt the delicate chemical balances necessary for clear cognition and emotional regulation. Despite these structural shifts, the brain possesses a remarkable, lifelong capacity for adaptation known as neuroplasticity. Even in the presence of atrophy, neurons retain the ability to forge new synaptic connections and reroute pathways around damaged areas. Research published in Nature Neuroscience suggests that the aging brain can recruit additional regions to complete tasks that once required only a single area, showing a high level of functional flexibility. This reorganization, sometimes called compensatory scaffolding, allows for continued learning and the refinement of complex skills well into the 9th decade of life. This resilience is further bolstered by a concept scientists call cognitive reserve, which functions like a biological savings account for the mind. Individuals who engage in lifelong learning, complex social interactions, and demanding mental activities tend to build more robust and redundant neural networks. These dense connections provide a structural buffer, allowing the brain to maintain high levels of function even when physical markers of aging or atrophy are present. It suggests that while the physical structure of the brain is subject to the inevitable laws of biology, the quality of our mental life is governed by a deeper, more personal history of intellectual curiosity. This relationship between the hardware of the brain and the software of experience defines the unique trajectory of how we age. This transition marks the boundary between fluid intelligence and its more enduring counterpart, crystallized intelligence. While fluid intelligence refers to the raw ability to process new information and solve abstract puzzles, crystallized intelligence is the vast library of knowledge and skills we accumulate over decades. The decline of fluid intelligence, which often begins as early as our 30s, is frequently offset by the steady growth of our vocabulary and general comprehension. Studies published in the journal Psychological Science suggest that certain linguistic abilities and social sensitivities do not even reach their peak until we are in our 60s or 70s. This growth in accumulated knowledge explains why pattern recognition often becomes more refined as we age, allowing the brain to see connections that a younger, faster mind might overlook. The aging brain becomes an expert at filtering out irrelevant data, focusing instead on the essential signals that inform sound judgment. Have you ever felt that a complex situation seemed much simpler to navigate now than it did a decade ago? This cognitive shift is supported by a neurological phenomenon known as the positivity effect, where the brain's amygdala shows a reduced response to negative images while maintaining a strong reaction to positive ones. This shift in emotional regulation allows older adults to maintain a more stable and optimistic outlook, prioritizing social harmony and meaningful connections over conflict. While the physical reaction time of the nervous system slows due to a gradual thinning of the protective myelin sheath around neurons, The brain compensates through increased contextual awareness. A slower response is often more accurate because it is filtered through a lifetime of precedents and outcomes, turning raw data into actionable wisdom. The brain effectively trades the high-energy volatility of youth for a more efficient, predictive model of the world. This internal reorganization ensures that even as the metabolic speed of our thoughts decreases, the quality of our conclusions remains high. This mental evolution relies heavily on the continuous stream of information provided by our surroundings, making the health of our sensory organs a critical factor in maintaining cognitive vitality. The way we perceive the world is not merely a product of thought, but a direct result of how our physical senses translate the environment into electrical signals. As the internal software of the mind adapts to its own changing architecture, the hardware of the senses undergoes its own remarkable transformation to keep the brain connected to the reality outside itself. The crystalline lens within the eye, once supple and clear, gradually accumulates cross-linked proteins that cause it to stiffen and lose its refractive grace. This physiological hardening, known as presbyopia, is why the fine print of a book eventually drifts further away from the face to find focus. By 2026, we have seen remarkable breakthroughs in accommodative intraocular lenses, yet the underlying biology remains a fundamental constant of the human experience. Deep within the retina, the density of photoreceptors also begins to thin with age. While we often notice changes in color perception, it is the rods, the cells responsible for vision in low-light environments, that typically decline more rapidly than the cones. This loss of sensitivity makes the transition from bright sunlight to a darkened room a much slower, more deliberate process for the aging mind. The auditory landscape undergoes a similar refinement as the decades pass. Inside the cochlea, the microscopic hair cells that vibrate in response to sound waves are subjected to years of mechanical stress and oxidative damage. These specialized cells do not naturally regenerate in humans, and their loss leads to presbycusis, a gradual fading of high-frequency registers. This isn't just a loss of volume, it is a loss of definition. Sibilant sounds like us or become increasingly indistinguishable, turning once clear sentences into a soft, indistinct murmur. This sensory thinning forces the brain to rely more heavily on context clues and visual signals to maintain the thread of a conversation, turning a simple social interaction into a demanding mental exercise. Our chemical senses of taste and smell also retreat into a more muted palate over time. The number of functional taste buds on the tongue decreases, and those that remain tend to atrophy, particularly affecting the perception of salty and sweet profiles. Have you ever noticed how the aromas of a familiar kitchen seem less overwhelming than they did in your youth? This occurs because the olfactory bulb, the neural structure responsible for processing scents, loses physical volume and fiber density. Since smell accounts for the vast majority of what we perceive as flavor, this dual decline can make the culinary world feel less vibrant, often leading to a natural, subconscious preference for more intense seasonings and textures. This systematic reduction in sensory input creates a significant increase in what scientists call cognitive load. When the brain receives noisy or incomplete data from the eyes and ears, it must work much harder to synthesize a coherent picture of reality. This extra effort consumes neural resources that would otherwise be used for memory storage or complex reasoning. To combat this, modern medicine has moved toward biological adaptations, such as gene therapies designed to protect surviving hair cells or advanced neural interfaces that sharpen signal clarity. These technologies, combined with the brain's natural plasticity and ability to reorganize its neural pathways, allow the aging individual to remain deeply connected to the environment, proving that the mind's desire for engagement is stronger than the limitations of its physical hardware. While the mind remains an expansive frontier, the physiological foundation of longevity rests upon the relentless rhythm of the cardiovascular system. As decades pass, the primary structural change involves a gradual loss of elasticity within the great vessels, a process often referred to as arteriosclerosis. The once supple walls of the aorta and its major branches begin to thicken and stiffen, primarily due to the cross-linking of collagen fibers and the degradation of elastin. This increased rigidity forces the heart to work harder to eject blood against higher resistance, which often leads to a subtle rise in systolic blood pressure. Over time, the left ventricle of the heart may thicken to compensate for this workload, a remodeling process that eventually impacts the heart's ability to relax and fill with blood between beats. The internal architecture of the heart also undergoes a quiet transformation, as the delicate valves that ensure one-way blood flow become thicker and less pliable. Calcification can occur at the base of these valves, particularly the aortic valve, which may slightly obstruct the pathway to the rest of the body or allow for minor backflow. Within the cardiac muscle itself, the number of pacemaker cells in the sinoatrial node, the body's natural metronome, decreases significantly by the time a person reaches their 8th decade. This loss of specialized cells can result in a slower resting heart rate and a reduced ability to rapidly increase the pulse during physical exertion. Consider for a moment the profound paradox of your existence, where the chaotic entropy of the universe meets the persistent, organized renewal of the biology of aging. It is a quiet phenomenon. Within the intricate architecture of your cells, a constant struggle unfolds between the inherent drive to maintain order and the inevitable accumulation of molecular noise. This process, formerly known as senescence, is not merely a linear decline or a simple wearing down of parts, but a highly coordinated biological symphony that responds to internal and external pressures. Every second, trillions of chemical reactions sustain your vitality, yet each one leaves behind a microscopic footprint, a tiny signature of time passing. This transformation reaches from the smallest subatomic particles and the fraying of DNA strands to the grand shifts in how societies are structured around longevity. We often rely on the steady rhythm of a calendar to measure our lives, but your body maintains a different ledger altogether. This is the critical distinction between your chronological age, the number of years you have occupied space on this planet, and your biological age, which reflects the actual physiological state of your tissues and organs. While the calendar moves at a fixed velocity, your biological clock can accelerate or decelerate based on a complex interplay of genetics, environment, and cellular metabolism. It is a fascinating reality that two people born on the same day can be decades apart in terms of cellular health. If you find these insights into our inner workings helpful for your bedtime listening, perhaps you might subscribe to join our quiet community of curious minds. The mechanism of aging is fundamentally rooted in the loss of information. As cells divide, the protective caps on the ends of chromosomes, called telomeres, gradually shorten like the burning wick of a candle. When these caps become too short, the cell enters a state of permanent arrest, no longer contributing to the repair of the body but instead secreting inflammatory signals that affect neighboring cells. This ripple effect contributes to the gradual thinning of skin and the slowing of wound healing. Interestingly, research published in the journal Nature suggests that certain organisms, like the Greenland shark, possess unique genetic adaptations that allow them to resist these processes for centuries. Do you remember a time when you first noticed a subtle change in your own physical endurance, or perhaps the way your skin felt in the morning? Such observations are our first personal encounters with the underlying mechanics of senescence. This journey into the biology of time reveals that we are not just victims of the clock, but active participants in a deeply complex and still largely mysterious evolutionary program designed to balance life and renewal. To understand why our cells eventually falter, we must look toward the fundamental laws of physics that govern the entire universe. The second law of thermodynamics dictates that entropy, or the measure of disorder, must always increase within a closed system. Life represents a temporary and defiant rebellion against this inevitable decay by constantly importing energy to maintain an internal state of high organization. Every heartbeat and every synaptic spark serves as a bulwark against the chaos that seeks to dissolve our structural integrity. This process is not free, as the body must burn fuel simply to stay the same, a concept often described in physics as maintaining a steady state far from equilibrium. The metabolic cost of this resistance is immense, requiring a constant flow of nutrients to power the molecular machinery of repair. Inside every cell, specialized enzymes act like a microscopic maintenance crew, patrolling the vast strands of DNA to fix breaks and errors caused by radiation or chemical stress. However, no mechanical or biological system achieves perfect efficiency, and a small fraction of the energy we consume produces harmful byproducts known as free radicals. These unstable molecules act like sparks flying from a forge, occasionally striking and damaging the very structures they were meant to support. Over several decades, these tiny, localized failures begin to aggregate into a significant burden of cellular noise that interferes with healthy function. As we move through the middle decades of life, the accumulation of these errors starts to outpace the body's ability to rectify them. Think of it as a long-term transcript that has been photocopied thousands of times. Eventually, the clarity of the original message is lost to the blur of repetition. This loss of information at the cellular level is a primary driver of the physical changes we associate with growing older. While the body is remarkably resilient, it is not designed for infinite durability, but rather for a specific window of peak performance. The joints, which act as the hinges of our mobility, face their own structural evolution as the water content of our cartilage begins to decrease. Cartilage is primarily composed of proteoglycans, molecules that attract and hold water to provide a cushioning effect. Over time, the synthesis of these molecules slows, causing the cartilage to become thinner and less resilient to mechanical stress. Simultaneously, the synovial fluid, the natural lubricant of our joints, may lose some of its viscosity. This lack of fluid lubrication can lead to the characteristic stiffness felt after long periods of inactivity, as the surfaces of the joints experience more direct friction. The skeleton is not a static object but a living, responsive organ that adheres strictly to the principle of mechanical loading. This use it or lose it reality is rooted in Wolff's Law, which states that bone grows or remodels in response to the forces placed upon it. When we engage in weight-bearing activities, the mechanical stress triggers the osteoblasts to strengthen the bone matrix. Without this regular stimulus, the body perceives the dense bone as an unnecessary energetic cost and allows the resorption process to continue unchecked. Maintaining this structural integrity through consistent resistance is perhaps the most vital component of long-term physical independence. The internal ecosystem of the human digestive tract undergoes a transformation as profound as the changes seen in our skeletal and muscular systems. Internal biodiversity often mirrors the external environment, and as the years pass, the variety of species living within the gastrointestinal tract begins to narrow. Research published in the journal Nature indicates that the elderly gut frequently lacks the microbial richness found in younger cohorts, a shift that is now recognized as a primary driver of the biological transition into later life. This loss of diversity is not merely a byproduct of time but a fundamental shift in how the body processes energy and regulates its immune responses. The intestinal wall serves as a highly selective barrier, but the structural integrity of this lining often degrades as we enter our 7th and 8th decades. When the tight junctions between cells begin to loosen, a phenomenon commonly known as increased intestinal permeability, or leaky gut, occurs. This allows metabolites and bacterial fragments to slip into the bloodstream, triggering a systemic immune response. This constant, low-grade infiltration contributes to a state known as inflammaging, where the body remains in a perpetual loop of irritation that consumes vital repair resources. Have you noticed how a simple change in your diet can alter your mental clarity for the rest of the day? This immediate connection is powered by the gut-brain axis, a bidirectional highway where microbial health dictates mood and cognitive sharpness. Certain bacteria are responsible for producing essential neurotransmitters like serotonin and gamma-aminobutyric acid, which influence emotional stability and sleep quality. As the microbiome shifts toward a less diverse state, the production of these chemicals can falter, potentially contributing to the cognitive fog and depressive symptoms often associated with advanced age. While beneficial populations like bifidobacteria often dwindle over time, opportunistic species such as proteobacteria tend to expand their footprint in the aging gut. These species thrive in the very inflammatory environments they help create, establishing a feedback loop that further destabilizes the internal ecosystem. This shift toward dysbiosis compromises the immune system's vigilance, making the body more susceptible to external pathogens while simultaneously weakening the internal defense mechanisms that protect against cellular damage. dietary fiber remains the most effective tool for maintaining microbiome stability, serving as the primary fuel source for beneficial bacteria. However, fiber intake frequently declines in older populations due to changes in appetite, digestion, or dental health. Consistently providing the gut with diverse plant-based fibers encourages the production of short-chain fatty acids like butyrate. These compounds are essential for strengthening the gut lining and suppressing the systemic inflammation that accelerates the aging process. By focusing on the maintenance of this microscopic landscape, it becomes possible to preserve the metabolic resilience required for a long and vibrant life. Please consider subscribing to continue this exploration of the human body's intricate systems as we uncover more about the science of longevity. The slowing of the internal clock is perhaps most visible in the steady decline of the basal metabolic rate. For most adults, this rate drops by roughly 2% every 10 years, a change driven primarily by the subtle loss of mitochondrial density within our cells. These tiny powerhouses become less efficient at converting nutrients into adenosine triphosphate, the primary energy currency of life. Consequently, the body begins to require less fuel for basic survival, yet the mechanisms that signal hunger do not always adjust with equal precision. This creates a physiological environment where the energy surplus is more easily stored rather than burned, a hallmark of the transition into later life. This shift is compounded by the emerging challenge of insulin resistance, where the body's cells become less responsive to the signals meant to regulate blood sugar. In a healthy state, insulin binds to receptors on the cell surface like a key in a lock, allowing glucose to enter and provide energy. As we age, those locks often become stiff or unresponsive, leading to higher levels of glucose circulating in the bloodstream. The pancreas attempts to compensate by producing more insulin, but this hyperinsulinemia can lead to a cycle of inflammation that accelerates the aging of the vascular system. This resistance is often linked to the reduction of GLUT4 transporters, which are the specific proteins responsible for moving sugar into muscle and fat cells. How do you personally find your body responds to changes in your diet or activity levels throughout the seasons? This metabolic friction often manifests as a redistribution of adipose tissue, moving away from the limbs and settling deep within the abdominal cavity. Unlike subcutaneous fat, which sits just beneath the skin, this visceral fat acts like an independent endocrine organ. It releases inflammatory signals known as cytokines that can impact the liver and heart directly. Researchers often refer to this phenomenon as inflammaging, a state where metabolic dysfunction and low-grade inflammation feed into one another, creating a self-sustaining loop that challenges cellular health. Simultaneously, the body experiences a marked reduction in the production of growth hormone and its partner, insulin-like growth factor 1. This decline, often termed somatopause, begins in early adulthood and continues throughout the later decades of life. Growth hormone is essential for maintaining bone density and lean muscle, and its absence makes the body more vulnerable to the physical wear and tear of daily existence. The liver, which produces the majority of our circulating IGF-1 in response to growth hormone signals, gradually slows its output, further reducing the regenerative capacity of our tissues. This metabolic trajectory is not a universal constant across the animal kingdom, however. While we face a predictable slowing of our chemical processes, other organisms have evolved entirely different relationships with energy. Some species maintain a nearly flat metabolic profile for their entire lives, showing no signs of insulin resistance even at advanced ages. By comparing our own insulin signaling and growth pathways with those of species that age differently, we can begin to see the specific trade-offs that nature has made in the design of the human body. This comparison reveals that while our metabolic decline is a standard part of the human experience, it is only one of many ways that life manages the passage of time. The mayfly represents the absolute extreme of this temporal spectrum, existing in its winged form for as little as 5 minutes or as long as 2 days depending on the specific species. Because their sole purpose in adulthood is reproduction, these insects lack functional mouths or digestive systems, essentially trading the ability to sustain life for the singular drive to pass it on. This fleeting existence serves as a stark reminder that biological success is not always measured in years, but in the successful handoff of genetic information. It is a peaceful realization to consider how some creatures pack an entire lifetime of purpose into the duration of a single afternoon while you enjoy these relaxing facts. Small mammals like the common house mouse navigate the world with a heart that beats up to 600 times per minute, a frantic pace that correlates with a lifespan of only 2 or 3 years. Biologists often point to the rate of living theory, which suggests that every organism is granted a roughly finite number of heartbeats, though modern research shows this is an oversimplification of complex cellular repair mechanisms. In these fast-paced lives, the mitochondria work at maximum capacity, generating the energy required for constant vigilance and rapid movement. This high metabolic throughput creates a significant amount of oxidative stress, which accumulates rapidly and triggers the signs of aging far sooner than in larger, slower-moving mammals. The concept of semiparity offers a haunting look at how evolution can explicitly schedule death as part of a life cycle, most notably seen in the Pacific salmon. After a grueling journey upstream to their ancestral spawning grounds, these fish experience a massive surge in corticosteroids that causes their internal organs to deteriorate at an accelerated rate. This isn't a failure of the body so much as a programmed biological sacrifice, ensuring that the parent does not compete with its offspring for limited resources in the freshwater environment. Do you remember the first time you realized that some living things are designed to end as soon as their greatest work is finished? Evolutionary pressure from predators plays a decisive role in setting the speed of a species' internal clock, often favoring those that mature and reproduce before they can be hunted. Species that face high extrinsic mortality, meaning they are likely to die from external causes, tend to age faster because nature sees no benefit in investing in long-term cellular maintenance. This contrasts sharply with the human trajectory, where our ancestors moved towards social cooperation and lower predation allowed for a much slower aging process. By extending our period of development and maturity, we have gained the luxury of decades, a biological rarity that sets us apart from the frantic, high-speed lives of the fast-agers in the animal kingdom. For those who find comfort in the steady rhythm of science for sleep, Understanding these rapid cycles helps us appreciate the deliberate, slow pace of our own biological clocks. Deep beneath the Arctic ice, the bowhead whale carries a genomic architecture that allows it to navigate the oceans for over 2 centuries. These massive mammals possess duplicated genes specifically related to cell cycle division and DNA repair, effectively outmaneuvering the cellular decay that plagues shorter-lived species. Research published in journals like Cell Reports has highlighted how these whales manage to suppress the deleterious mutations that typically accumulate with age. While we often associate size with longevity, the Greenland shark takes this principle to an extreme, potentially reaching ages of 400 years or more in the frigid, high-pressure depths. Their incredibly slow metabolic rate means they may not even reach sexual maturity until they have lived for a century and a half. This glacial pace of life suggests that metabolic conservation is a primary defense against the oxidative stress that accumulates in warmer-blooded creatures. Contrasting these giants is the naked mole rat, a subterranean rodent that defies nearly every rule of mammalian senescence. Despite being the size of a common mouse, which typically lives only 3 years, the naked mole rat can survive for over 3 decades in its oxygen-deprived tunnels. Their resistance to cancer is nearly absolute, a feat attributed to high molecular mass hyaluronan, which triggers early contact inhibition in their cells. This biological mechanism ensures that cells stop dividing long before they can form a tumor. They also exhibit a curious lack of pain sensitivity to acid and heat, showing us that longevity often comes with a suite of radical physiological adaptations. Do you ever find yourself wondering how our own medical technology might one day mimic these natural defenses as you drift towards sleep? This disparity between size and cancer risk leads us to the observation known as Peto's paradox. Statistically, an organism with more cells should have a higher probability of a stray mutation turning into a malignancy, yet whales have significantly lower cancer rates than humans or mice. Large animals have evolved superior tumor suppressor mechanisms, such as multiple copies of the p53 gene, which acts as a guardian of the genome. By investing in these redundant safety systems, these species prove that biological complexity does not have to be a liability. The secret to these long lives resides in the efficiency of the molecular machinery responsible for fixing broken DNA strands. Species like the bowhead whale have more efficient double-strand break repair pathways, preventing the genomic instability that characterizes human aging. These mechanisms involve proteins like SIRT6, which coordinates the repair process and maintains the integrity of the chromatin structure. As we observe these master survivors, the line between extreme longevity and biological immortality begins to blur. Some organisms, like the tiny hydra, appear to lack a discernible aging process altogether, possessing stem cells that constantly renew their entire body. This ability to reset the biological clock suggests that death, at a cellular level, may not be an inevitability of nature but rather a programmed limitation that some life forms have learned to bypass. Understanding these pathways brings us closer to a future where aging is a condition to be managed rather than an inescapable fate. Nature harbors creatures that suggest biological immortality is not just a myth but a documented reality within our oceans. These organisms challenge our core definitions of aging. The hydra, a small freshwater invertebrate, possesses a unique population of stem cells that constantly renew themselves, effectively preventing the organism from showing any signs of senescence. If you find these relaxing facts about biology helpful for your bedtime listening, feel free to subscribe for more science for sleep. Observations published in the journal Nature have shown that hydra do not experience an increase in mortality rates as they get older, essentially making them biologically ageless. Their entire body is composed of 3 distinct stem cell lineages that replace every cell in the creature approximately every 20 days. This constant turnover means that a hydra you observe today is, at a cellular level, entirely different from the one you saw a month ago. Another remarkable example is found in the deep sea with the species Turritopsis dohrnii, commonly known as the immortal jellyfish. When this creature faces physical trauma or environmental stress, it undergoes a rare process called transdifferentiation. During this cycle, the adult medusa cells transform back into specialized cells of a juvenile polyp stage, effectively hitting a biological reset button. This allows the jellyfish to bypass death and begin its life cycle anew as a colonial polyp on the seafloor. Do you find yourself more drawn to the mysteries of the deep ocean or the vastness of the stars when you are trying to fall asleep? The concept of negligible senescence extends to larger animals as well, such as certain species of lobsters and tortoises. Lobsters are particularly notable because they express high levels of the enzyme telomerase throughout their adult tissues, which maintains the length of their telomeres during every cell division. While most mammals eventually succumb to the degradation of their genetic protective caps, lobsters continue to grow and remain fertile until external factors, like disease or predation, intervene. Similarly, the Galapagos tortoise exhibits a physiological resilience that defies the standard mammalian timeline of decay. Research into their genomes has identified gene variants associated with DNA repair and immune response that are far more robust than those found in humans. Despite these incredible mechanisms existing in the natural world, humans are currently prevented from utilizing them due to the sheer complexity of our specialized tissues. In complex mammals, high telomerase activity is often linked to the uncontrolled cell growth seen in oncology, making a simple immortality switch dangerous for our survival. Furthermore, our most vital information is stored in the intricate synaptic connections of the brain, which cannot be easily reset or replaced without losing the continuity of the individual. Even with these barriers, the existence of these species proves that death is not a thermodynamic necessity for all living matter. Biological immortality remains a rare but functional reality, existing as an alternative evolutionary strategy that prioritizes cellular maintenance over rapid reproduction. Understanding how these organisms maintain their genomic integrity offers a blueprint for future medicine, where we might one day borrow their secrets to extend the period of healthy human life. As we continue to decode these ancient survival strategies, the line between an inevitable end and a manageable biological process becomes increasingly blurred. The quest for longevity often leads to specific geographic pockets where the centenarian population defies global averages by a significant margin. These regions, famously mapped by researchers as blue zones, provide a natural experiment in human aging and environmental interaction. In the mountainous Barbagia region of Sardinia, Italy, researchers discovered the highest concentration of male centenarians in the world. This demographic anomaly is largely attributed to a combination of genetic isolation and a lifestyle involving high daily energy expenditure through traditional shepherding practices across rugged terrain. The archipelago of Okinawa, Japan, has long been celebrated for having the longest disability-free life expectancy, particularly among women. This longevity is supported by Moai, a social support network that provides lifelong financial and emotional security. Do you remember a time when a simple conversation with a friend felt like it lifted a heavy weight from your mind? This emotional buffering reduces chronic cortisol levels, which is a known driver of cellular senescence and systemic inflammation. Beyond social ties, the Okinawan practice of hara-hachi-bu instructs individuals to stop eating when they are 80% full, naturally moderating caloric intake. In Nicoya, Costa Rica, the local water supply is naturally enriched with high levels of calcium and magnesium, which supports bone density and cardiovascular function in the elderly. These physical benefits are paired with a strong plan de vida, or soul-deep purpose, which keeps the aging brain cognitively engaged and resilient. Similarly, the inhabitants of Icaria, Greece, exhibit incredibly low rates of middle-age mortality and dementia. This Greek island's secret appears to lie in the consumption of antioxidant-rich herbal teas and the preservation of the midday nap, which helps regulate blood pressure and stress hormones. Across these diverse cultures, the Power 9 principles emerge as a cohesive framework for longevity that transcends individual genetics. Science suggests that a plant-slant diet, favoring legumes over processed meats, reduces the intake of specific amino acids that can overstimulate growth pathways. Natural movement, rather than episodic high-intensity exercise, ensures a steady metabolic rate and prevents the progressive stiffening of the vascular system. Even the Seventh-day Adventists in Loma Linda, California, demonstrate that a shared sense of community and a diet focused on whole grains and nuts can add nearly a decade of healthy life compared to their immediate neighbors. By the current year of 2026, we are witnessing the unfortunate erosion of these longevity havens as globalized food systems and sedentary digital habits reach even the most remote villages. In Okinawa, younger generations now face metabolic challenges similar to those in Western nations, leading to a decline in the traditional longevity advantage once enjoyed by their ancestors. This transition highlights that geography is not a permanent shield, but rather a container for habits that must be actively preserved. As these traditional environments fade, modern biology is looking to replicate their physiological effects through more precise metabolic interventions. One of the most consistent findings from these regions is the habit of caloric moderation, which brings us to the profound biological impact of how much energy we consume. The foundation of our understanding rests heavily on longitudinal studies involving rhesus monkeys, our close evolutionary relatives. Decades of research at the University of Wisconsin-Madison demonstrated that monkeys on a restricted diet lived significantly longer and appeared biologically younger than those fed a standard diet. While a parallel study by the National Institute on Aging initially showed less dramatic results, a collaborative analysis eventually revealed that the source of calories, specifically the avoidance of processed sugars, played a critical role in how fasting influences the aging process. This breakthrough highlighted that the metabolic benefits of restriction are most potent when paired with high-quality, nutrient-dense intake rather than just raw caloric reduction. When the body senses a drop in available energy, it flips a metabolic switch from growth to preservation. This shift is mediated by the inhibition of the mTOR pathway, a central regulator of cell growth and protein synthesis. Under normal conditions of plenty, mTOR promotes cell proliferation, which is essential for development but can accelerate the aging process if left unchecked throughout adulthood. By suppressing this pathway through fasting, we permit the cell to redirect its limited resources away from building new structures and toward repairing existing ones. This transition is a fundamental survival mechanism, evolved to keep organisms viable during periods where food was naturally scarce. This suppression works in tandem with the activation of sirtuins, a group of 7 specialized enzymes that respond to cellular stress. Sirtuins serve as the molecular link between nutrient availability and gene expression, effectively quieting the noise in our genetic code that accumulates over time. They are particularly adept at protecting the telomeres, the protective caps at the ends of our chromosomes that shorten every time a cell divides. Have you ever noticed how your mental focus seems to sharpen slightly when you haven't eaten for several hours? This clarity is often a byproduct of the body entering this heightened state of physiological readiness and resource management. It is important to recognize that these benefits do not require the extreme deprivation associated with starvation. True starvation depletes the body's essential reserves, leading to systemic failure and muscle wasting, whereas caloric restriction and fasting mimicking diets are carefully calibrated to maintain nutritional adequacy. These mimicking protocols involve a strategic reduction in protein and carbohydrates for short, controlled periods, inducing the same protective molecular responses without the physical toll of a prolonged total fast. This allows the modern individual to access ancient survival pathways while still maintaining the energy required for daily life. The scarcity of nutrients serves as an evolutionary signal that it is time for cellular repair rather than reproduction. This triggers autophagy, a sophisticated recycling system where cells consume their own damaged components to generate energy. By dismantling misfolded proteins and malfunctioning mitochondria, autophagy prevents the cellular clutter that leads to systemic inflammation and chronic organ decline. This deep cleaning mechanism is perhaps the most potent tool the body possesses to maintain youthful function at the microscopic level. Through these precise metabolic interventions, we are learning to command our cells to prioritize longevity over immediate growth, effectively slowing the ticking clock of our biological age. Muscle tissue acts as the body's largest endocrine organ, a discovery that has fundamentally changed how we view the physiology of aging. When muscle fibers contract under load, They release a cascade of signaling proteins known as myokines into the bloodstream. These molecules serve as chemical messengers that communicate directly with the brain, the liver, and even our adipose tissue to regulate systemic inflammation. One specific myokine, known as irisin, has been shown to cross the blood-brain barrier, where it helps protect neurons from the degenerative processes associated with cognitive decline. The mechanical stress of movement also triggers a process called mitochondrial biogenesis. Within our cells, specialized pathways like PGC1-alpha are activated, instructing the body to manufacture entirely new mitochondria to meet the increased energy demand. This expansion of the cellular power grid is vital because aging often involves a decline in mitochondrial density and efficiency. By maintaining a robust population of these organelles, we reduce the leakage of reactive oxygen species that typically accelerates cellular damage over time. Do you remember the first time you felt the distinct, heavy glow of physical exhaustion after a long day of movement? Resistance training offers a unique defense against the structural degradation of the skeletal system. Through the principle of Wolff's law, the mechanical strain placed on bones during lifting stimulates osteoblasts to lay down new mineral matrix, effectively reversing the age-related loss of bone density. Simultaneously, this process enhances insulin sensitivity by increasing the expression of GLUT4 transporters in the muscle cell membranes. Because skeletal muscle is the primary site for glucose disposal, maintaining muscle mass acts as a metabolic buffer against the chronic high blood sugar that leads to advanced glycation end products. In the landscape of the brain, exercise facilitates the release of brain-derived neurotrophic factor, or BDNF. Often described by researchers as miracle grow for the mind, BDNF supports the survival of existing neurons while encouraging the growth of new ones in the hippocampus, the region responsible for memory and spatial navigation. This neuroplasticity is a cornerstone of cognitive longevity, ensuring that the brain remains adaptable even as the decades pass. Physiological exertion serves as a profound systemic signal for survival and maintenance. From an evolutionary perspective, intense activity signaled to our ancestors' bodies that the environment was challenging and that every system needed to operate at peak efficiency to ensure survival. Conversely, prolonged physical inactivity may inadvertently signal to the body that high-level maintenance is no longer necessary, leading to the gradual pruning of metabolic and structural capabilities. Through exercise, we are essentially hacking these ancient biological pathways to maintain a state of high-functioning vitality. These relaxing facts about our internal chemistry remind us that movement is not just about performance, but about the very preservation of our biological integrity. As you listen to this science for sleep, consider how these microscopic shifts are happening right now, even in your moments of stillness. Moving into the deeper structures of our anatomy, we find that the influence of exercise extends even to the way our DNA is packaged and protected. Deep within the architecture of the aging brain, a silent plumbing system known as the glymphatic pathway works tirelessly to maintain cognitive health. This network serves as a specialized waste clearance system, utilizing a macroscopic fluid transport mechanism to flush out metabolic byproducts that accumulate during our waking hours. While the rest of the body relies on the lymphatic system to manage cellular debris, the brain, shielded by the blood-brain barrier, utilizes this unique lymphatic process to ensure its internal environment remains pristine. It is a masterpiece of biological engineering that relies heavily on the structural changes occurring while we rest. During the stages of deep, slow-wave sleep, the interstitial space between our neurons actually expands by up to 60%. This expansion allows cerebrospinal fluid to surge through the brain tissue with significantly less resistance, effectively washing away the day's molecular waste. This nocturnal rinse is essential for the removal of amyloid beta, a protein fragment that, if left to aggregate, is a primary hallmark of neurodegenerative decline. Research has shown that even a single night of significant sleep deprivation can lead to a measurable increase in amyloid beta levels within the thalamus and hippocampus. Have you ever noticed how a night of poor rest leaves your thoughts feeling physically heavy or clouded the next morning? As the body ages, the efficiency of this lymphatic drainage often begins to falter, partly due to the natural stiffening of arterial walls which help drive the fluid flow. Furthermore, the aging process typically involves a gradual shift in our internal circadian rhythms, often resulting in advanced sleep phase syndrome, where the desire for sleep occurs earlier in the evening. This shift is accompanied by a decline in the production of melatonin, the hormone secreted by the pineal gland that signals the body it is time to transition into a restorative state. Without sufficient melatonin signaling, the transition into the deepest, most lymphatically active stages of sleep becomes increasingly fragmented. The architecture of sleep itself undergoes a profound transformation as the decades pass, with a notable reduction in the duration of rapid eye movement or REM cycles. While deep sleep handles the physical cleaning of the brain, REM and lighter stages of sleep are critical for the complex process of memory consolidation. During these phases, the brain replays and strengthens the neural connections formed during the day, integrating new information into our long-term narrative. When sleep is interrupted or shortened, this consolidation process is truncated, making it harder for the aging brain to bridge the gap between short-term acquisition and permanent storage. Interestingly, the quality of our sleep and the health of our lymphatic system exist in a reciprocal relationship, where poor sleep accelerates aging and aging further disrupts sleep. This feedback loop underscores the importance of maintaining consistent sleep hygiene to protect the brain's physical integrity over time. Scientists now understand that the brain does not simply shut down at night. It enters a period of intense metabolic housekeeping. By prioritizing these quiet hours, we are essentially providing the brain with the resources it needs to repair DNA damage and clear the toxic proteins that threaten our long-term clarity. This hidden cycle of renewal is perhaps the most potent tool we have for preserving the essence of who we are as the years continue to unfold. While those nocturnal cycles of repair offer a vital sanctuary for the brain, our waking hours are often dominated by a physiological system that can inadvertently accelerate the clock. The hypothalamic pituitary adrenal axis, or HPA axis, serves as the body's primary command center for managing external pressures. When the brain perceives a threat, this axis triggers the release of cortisol, a steroid hormone that reshapes our metabolism to handle immediate danger. In short bursts, cortisol is protective, but the modern condition of chronic, low-level stress keeps this system perpetually engaged. Over time, elevated cortisol levels act like a slow-moving corrosive, particularly within the hippocampus, where it can inhibit the birth of new neurons and contribute to the thinning of brain tissue. This chemical persistence reaches deep into our genetic architecture, specifically targeting the telomeres we discussed earlier. Research published in Proceedings of the National Academy of Sciences has demonstrated that individuals experiencing chronic psychological distress often possess significantly shorter telomeres than their less stressed peers. It appears that the biochemical environment created by constant worry may prematurely exhaust the enzyme telomerase, effectively forcing our cells into an early state of senescence. This isn't just a matter of feeling tired, it is the physical manifestation of life's burdens written into the very ends of our chromosomes. Beyond the genetic level, our internal defense systems also pay a steep price for this heightened state of alertness. The thymus, a specialized gland responsible for training the T cells that fight off infections, is remarkably sensitive to the presence of stress hormones. Under the weight of long-term cortisol exposure, the thymus undergoes a process called involution, shrinking and losing its capacity to produce a diverse array of immune cells. This creates a state of immunosenescence, where the body becomes less adept at recognizing new pathogens or clearing out the damaged cells that can lead to systemic inflammation. However, the relationship between tension and aging is more nuanced than it first appears. Biologists often distinguish between distress, which is the overwhelming, chronic pressure that wears us down, and eustress, which is the positive, manageable challenge that keeps us sharp. While distress drains our biological reserves, eustress can actually stimulate cellular resilience. This leads us to the fascinating concept of hormesis, the idea that small, controlled amounts of stress can act as a catalyst for longevity. When we expose our systems to brief, intense stimuli, we don't just endure the pressure. We trigger an overcompensation mechanism that leaves our cells stronger than they were before the challenge began. This biological principle, known as hormesis, describes a biphasic dose-response relationship where a substance or environmental factor that is harmful in high doses becomes incredibly beneficial at low levels. It suggests that our bodies are not designed to exist in a state of perpetual comfort, as total ease often leads to biological stagnation and the gradual atrophy of our internal repair systems. When we intentionally step outside our comfort zones, we provide the necessary signals for our cells to maintain their youthful vigor. When you step into the intense heat of a sauna, your core temperature rises, signaling a state of emergency to your cells and prompting the rapid synthesis of heat shock proteins. These specialized molecules serve as vital quality control agents, patrolling the cellular environment to refold damaged proteins and prevent the toxic aggregates often associated with neurodegenerative decline. Long-term studies, such as those conducted in Finland, have shown that this specific type of thermal stress can significantly lower the risk of cardiovascular events by improving arterial compliance and reducing systemic inflammation. In sharp contrast, plunging into cold water activates a different suite of protective mechanisms, specifically cold shock proteins like RBM3, which have been shown in laboratory settings to protect neurons and maintain synaptic connections. This sudden drop in temperature also stimulates the recruitment and activation of brown adipose tissue, a highly metabolic form of fat that is rich in mitochondria and designed to burn energy specifically for heat production. Have you ever noticed how the sharp sting of a cold shower seems to leave you feeling more grounded and mentally clear once the initial shock fades away? Even the vegetables we consume play a role in this stress response dance through a process called xenohormesis. Many of the most healthful compounds in plants, such as the sulforaphane found in broccoli or the polyphenol in dark berries, are actually mild toxins the plant produces to ward off pests. When we ingest these phytochemicals, they trigger the NRF2 pathway, a master regulator that coordinates the expression of over 200 cytoprotective genes, effectively turning on our internal antioxidant pharmacy. By viewing resilience as a biological muscle, we begin to see that aging is not just a process of inevitable wear and tear, but a gradual loss of the ability to respond effectively to environmental challenges. Regularly dipping into these controlled stressors keeps our cellular machinery oiled and attentive, ensuring that the ancient systems responsible for repair and maintenance remain online and efficient throughout the decades. Subscribing to this lifestyle of gentle, rhythmic challenge helps ensure your biological age stays well below your chronological years. The shifting landscape of modern medicine has moved beyond merely treating the symptoms of decay, pivoting toward a class of compounds known as geroprotectors. These pharmacological agents are designed to intervene directly in the biological aging process, aiming to extend the period of life spent in good health rather than simply lengthening the duration of old age. Scientists distinguish these from traditional medicines by their ability to target the fundamental hallmarks of aging across various tissue types simultaneously. One of the most heavily researched molecules in this category is rapamycin, a compound originally discovered in the soil of Easter Island. Within our cells, rapamycin acts as a potent inhibitor of the mechanistic target of rapamycin, or mTOR, which functions as a primary nutrient sensor. When mTOR is active, the cell focuses on growth and protein synthesis. When it is inhibited, the cell shifts its energy toward internal cleanup processes like autophagy. This cellular recycling is crucial for removing damaged proteins and organelles that otherwise accumulate over time. While rapamycin is FDA approved for preventing organ transplant rejection at high doses, the longevity community focuses on low-dose, intermittent protocols. Research in mice has consistently shown lifespan extension even when the drug is started late in life, though human application remains cautious due to potential side effects like mouth sores or altered glucose metabolism. Do you remember the first time you felt a genuine sense of wonder about how the tiny components inside your body actually communicate with one another? Parallel to rapamycin is metformin, a common medication for type 2 diabetes derived from the French lilac plant. Its reputation as a longevity tool stems from observational data suggesting that diabetics on metformin often outlive non-diabetics who are otherwise healthy. This led to the landmark TAME trial, or Targeting Aging with Metformin, which represents a shift in regulatory thinking by treating aging itself as a modifiable condition rather than an inevitability. Metformin is believed to work by activating AMPK, an enzyme that mimics the beneficial effects of low-energy states, essentially telling the body to optimize its metabolic efficiency. The quest for cellular energy often leads to the discussion of nicotinamide adenine dinucleotide, or NAD+, a coenzyme found in every living cell. NAD+ is essential for the function of sirtuins, a family of proteins that repair damaged DNA and regulate circadian rhythms. Because NAD+ levels naturally decline as we age, precursors like nicotinamide riboside and nicotinamide mononucleotide have become staples of the longevity conversation. These precursors aim to replenish the cellular fuel tank, supporting mitochondrial health and systemic resilience. However, the transition from laboratory success to human longevity remains complex, as biological systems are remarkably adept at maintaining equilibrium. Taking a pill to boost a specific pathway can sometimes trigger compensatory mechanisms that negate the benefits or cause unintended stress on the liver and kidneys. These substances are increasingly viewed as specialized tools rather than magic pills, requiring precise timing and dosage to be effective without causing harm. The current consensus in 2026 emphasizes that no pharmacological intervention can fully compensate for a lack of sleep or poor nutrition. Instead, these compounds are seen as the last mile of longevity, small but significant adjustments that can sharpen the biological edge provided by a foundational lifestyle. Science has shown that while we can influence the rate of our decline, the most powerful interventions remain those that support the body's innate desire for balance and repair. Integrating these molecular tools requires a nuanced understanding of one's own unique biochemistry, as the future of pharmacology is moving toward personalized, data-driven protocols that respect the complexity of the human form. Within the delicate architecture of our tissues, some cells enter a state of permanent arrest known as senescence, refusing to die while secreting inflammatory signals that degrade their neighbors. These lingering entities, often called zombie cells, accumulate over decades and drive many of the chronic conditions we once viewed as inevitable. Senolytics represent a sophisticated pharmacological intervention designed specifically to identify and eliminate these dysfunctional cells without harming the healthy ones surrounding them. By targeting the survival pathways that senescent cells use to evade programmed cell death, these drugs allow the body to naturally clear away the cellular debris that clogs our biological systems. The most documented approach in recent years involves the combination of dasatinib, a tyrosine kinase inhibitor, and quercetin, a naturally occurring polyphenol found in many fruits and vegetables. Clinical trials throughout 2024 and 2025 demonstrated that this pairing could significantly reduce the senescent cell burden in the kidneys and adipose tissue of elderly participants. This reduction in the senescence-associated secretory phenotype, or SASP, effectively lowers the systemic background noise of inflammation. When the inflammatory signals are silenced, the surrounding healthy cells can function with greater efficiency, often leading to improved physical function and mobility. Have you ever noticed how a deep, uninterrupted sleep seems to make your memory feel sharper and your body more resilient the following morning? Beyond the removal of damaged cells, The biotechnology landscape of 2026 has ventured into the realm of cellular restoration through partial reprogramming. This technique utilizes the Yamanaka factors, a set of 4 specific genes that earned Shinya Yamanaka the Nobel Prize for their ability to revert a mature cell back into a pluripotent stem cell state. The contemporary challenge lies in the duration of exposure. If these factors are expressed for too long, cells lose their specialized identity entirely, which can lead to the formation of disorganized tissue masses known as teratomas. To mitigate this risk, researchers have refined transient reprogramming protocols that provide just enough stimulus to reset the cell's epigenetic clock without erasing its functional memory. This delicate balance allows a heart cell or a neuron to remain exactly what it is while regaining the molecular profile and energy production levels of a much younger version of itself. We are currently observing how these gene therapies might be delivered via lipid nanoparticles or mRNA technologies to target specific organs, bypassing the need for systemic genetic alteration. This shift from broad-spectrum drugs to targeted genetic instructions marks the definitive frontier of current longevity science. By merging the precision of senolytics with the transformative potential of epigenetic resetting, medicine is moving away from simply managing the symptoms of decay toward addressing the fundamental mechanics of biological time. It is an era defined by the transition from merely repairing damage to instructing the body to remember its own youthful blueprint, creating a future where the biological age of our tissues no longer has to match the chronological count of the years we have lived. Ovarian aging presents a unique biological trajectory because it occurs at a pace far exceeding the aging of other organ systems. While the rest of the body maintains its structural integrity into the 6th or 7th decade, the female reproductive system often undergoes a programmed cessation of function by the late 40s or early 50s. This transition, known as menopause, is characterized by the depletion of the primordial follicle pool, which serves as the finite reservoir of potential life. As these follicles vanish, the production of essential hormones like estrogen and progesterone begins to fluctuate and eventually collapses. This hormonal shift is not merely a reproductive event but a systemic transformation that impacts the entire female physiology. Estrogen, for instance, acts as a critical regulator of bone density and cardiovascular health, acting as a protective shield against arterial stiffness and skeletal fragility. When these levels drop, the risk of osteoporosis and heart disease increases, highlighting how deeply our reproductive clocks are intertwined with our longevity. In contrast, the male experience of reproductive aging, sometimes referred to as andropause, follows a more gradual and subtle slope. Testosterone levels typically decline by about 1% per year starting after age 30, yet many men maintain their fertility well into their twilight years. This slow tapering lacks the definitive, abrupt end seen in females, though it still influences muscle mass, cognitive clarity, and metabolic rate. From an evolutionary standpoint, the existence of a long post-reproductive lifespan in humans is a profound mystery that sets us apart from almost all other primates. Most animals in the wild die shortly after they lose the ability to reproduce, yet human females often live for decades beyond their fertile years. This anomaly is often explained through the grandmother hypothesis. which suggests that the survival of our species was bolstered by elder females who no longer bore their own children. By redirecting their energy toward the care and provisioning of their grandchildren, these women increased the survival rates of their descendants and allowed their daughters to have more children in shorter intervals. This cooperative breeding strategy effectively turned the cessation of fertility into a biological advantage for the tribe. Have you ever considered how your own family's history might have been shaped by the wisdom and support of a previous generation during your childhood? This transition from personal reproduction to ancestral investment suggests that our later years are not a period of biological decline, but a specialized stage of human life designed for the stability of the collective. The legacy of our reproductive history is thus written into the very architecture of our social structures, ensuring that life continues to flourish even as our individual clocks slow down. Evolutionary biologists observe a rare phenomenon in the human species: the extended post-reproductive lifespan of females, a concept widely known as the grandmother hypothesis. This theory posits that the survival of our ancestors was significantly bolstered when older women transitioned from bearing their own children to investing in the care of their grandchildren. By providing this essential support, grandmothers increased the survival rates of the next generation and allowed younger mothers to have children at shorter intervals. This shift created a unique evolutionary feedback loop where longevity became a survival trait, effectively weaving the presence of elders into the very fabric of human success. Do you remember a specific story or piece of advice passed down to you by an older family member? The presence of these non-reproductive adults steered our species toward a longer life, as those who lived longer provided more stability and knowledge to their kin. In prehistoric societies, the value of an elder was measured by their role as a living library. They held the collective memory of the tribe, identifying which roots were safe to eat during a drought or how to track weather patterns across the seasons. This social utility transformed the aging process from a period of decline into a specialized stage of cultural transmission. Our biological systems are still wired for this deep interconnectedness, which makes the modern epidemic of isolation particularly dangerous. We now recognize that social isolation is among the most significant predictors of mortality, often rivaling the risks posed by long-term tobacco use. When an individual lacks consistent social interaction, the brain perceives this as a threat to survival, triggering a cascade of stress hormones like cortisol. This persistent state of high alert causes systemic inflammation, which accelerates the aging of the heart and the brain. Researchers frequently describe loneliness as a biological toxin because of how it degrades cellular health. Recent meta-analyses from late 2025 indicate that the inflammatory markers found in chronically lonely individuals are remarkably similar to those found in people suffering from chronic physical ailments. This reveals that our longevity is not just a matter of genetics or nutrition, but is deeply dependent on our proximity to others. Our cells seem to know when we are alone, and in that silence, they begin to fail more rapidly. The evolutionary legacy that once rewarded us for living long enough to help others now punishes us when that social contract is broken, proving that aging is a communal experience rather than a solitary one. If the architecture of our society continues to move away from intergenerational living, we risk losing the very biological protection that allowed our species to thrive in the first place. Within this biological framework lies a striking contradiction known to researchers as the paradox of well-being. While physical health often follows a downward trajectory in later years, subjective happiness frequently moves in the opposite direction. Data collected across more than 130 countries confirms the existence of a U-curve of happiness, where life satisfaction typically bottoms out in the mid-40s before beginning a steady, upward climb. This suggests that the psychological experience of aging is not one of steady decline, but rather an ascent toward greater emotional stability. Psychologist Laura Carstensen developed the socio-emotional selectivity theory to explain why this shift occurs as our internal clock winds down. When people perceive their time as expansive, they prioritize long-term goals, such as gathering information or building professional networks. As the temporal horizon nears, the brain shifts its focus toward present-oriented satisfaction and emotionally meaningful experiences. This is why many older adults naturally prune their social circles, choosing to invest deeply in a few high-quality relationships rather than maintaining a wide web of acquaintances. Can you recall a moment in your youth when a minor social slight felt like an earth-shattering event, only to realize now how quickly those feelings faded? This change is rooted in the positivity effect, a cognitive bias where older brains tend to prioritize positive information over negative stimuli. Functional MRI scans have shown that the amygdala, which processes emotions, becomes less reactive to negative images as we age, while maintaining its responsiveness to positive ones. This neurological filtering system helps protect the mind from the stressors that often plague younger, more reactive individuals. This emotional regulation is not merely a passive result of aging but a sophisticated cognitive skill that improves over decades. Older adults are generally more adept at affective forecasting, which is the ability to predict how an event will make them feel, allowing them to avoid unnecessary conflict. They also exhibit a higher capacity for complex emotional reasoning, often viewing social disagreements with a nuanced perspective that younger people might miss. This psychological resilience acts as a vital counterbalance to the physiological changes occurring in the cells. The transition from a future-oriented mindset to a present-oriented one changes the very nature of what brings us joy. Instead of seeking the dopamine rush of novelty or achievement, the aging brain finds deeper contentment in the oxytocin-rich experiences of connection and legacy. It is a biological irony that the period of life often associated with loss is frequently reported as the most peaceful. By shifting our focus from what we might become to what we currently are, the mind creates a sanctuary of well-being that remains remarkably independent of physical vitality. The science of aging continues to reveal that while our bodies are designed to eventually fail, our minds are wired to find increasing value in the time that remains. If you find these insights into the human journey helpful, perhaps you might consider subscribing to stay updated on our latest explorations of the natural world. This emotional maturation ensures that the final chapters of life are defined not by the shadow of the end, but by the clarity of the present. The tapestry of aging is not woven from a single thread, but is instead a complex intersection of countless biological mechanisms acting in concert across the lifespan. It represents a vast, multifaceted collection of processes, ranging from the gradual shortening of protective telomeres to the accumulation of zombie senescent cells that linger in our tissues and drive chronic inflammation. In our current landscape of early 2026, we have moved firmly beyond the historical view of aging as an inevitable metaphysical fate. Instead, we treat it as a series of specific, quantifiable biological challenges that can be decoded, measured, and eventually addressed through targeted intervention. This shift marks one of the most significant transitions in the history of medicine as we move from treating the symptoms of age-related disease to understanding the underlying drivers of the aging process itself. This new paradigm has brought the concept of longevity escape velocity into the mainstream scientific dialogue. The idea suggests a future point where medical breakthroughs add more than 12 months to the average human life expectancy for every calendar year that passes, theoretically allowing for an indefinite extension of health span. While the prospect is tantalizing, critical voices in the scientific community remind us that the human body is an incredibly intricate system. Solving the puzzle of protein misfolding or mitochondrial decay in one organ does not automatically resolve the damage occurring in another. The quest for longevity must be tempered with the reality of our biological complexity, ensuring that we do not merely add years to life, but truly add vibrant life to those years. Focusing exclusively on the technical extension of time can sometimes obscure the unique value inherent in the aging process itself. There is a specific form of emotional intelligence and intellectual perspective that only matures through the sustained accumulation of decades of lived experience. This slow-cooked wisdom is a vital component of the human narrative, providing a grounding influence that younger generations rely upon for stability and context. Is there a specific insight or perspective you have gained with time that you feel you simply could not have understood in your youth? If you find these explorations into the science of our existence meaningful, perhaps you might consider subscribing to stay connected with our ongoing journey through the wonders of the natural world. Our understanding of time and biology continues to evolve, yet the fundamental curiosity that drives us remains a constant. As we continue to unlock the secrets of our own selves, we find that every discovery only adds to the beauty of the human experience. The pursuit of science is a testament to our desire to understand the finite while reaching for the infinite, leaving us with a deeper appreciation for every moment we are given.